Okay, it's a pleasure to welcome one of the world's experts on cell cycle and chromosome organization, Professor Bill Earnshaw from the University of Edinburgh. Bill's lab is focused on cell cycle control of chromosome organization, and he's focused heavily on the topic he's going to tell us about today, metaphase mitotic reorganization of chromosomes. He also works on cell death, apoptotic reorganization of chromosomes. And Bill was originally from Maine and went to college in Maine and then went to MIT to do his PhD with Jonathan King. And there he did a variety of projects, including some of early work elucidating how DNA is folded in viral capsids. He then went on to a series of postdocs, one in Cambridge in the group of Aaron Klug, and another postdoc with Ulrich Lemley at the University of Geneva. And he wrote a number of, with Lemley, a number of very influential works on mitotic chromosome organization. He then became a faculty member at Johns Hopkins and rose through the ranks to full professor there. And in 1996, he moved to the University of Edinburgh in Scotland and now is a principal research fellow of the Wellcome Trust. And for those of you that don't know, that's something like being an HHMI lab director here. So some of the things his lab has done, he's developed the first in vitro system to study programmed cell death. He really has established, his work with Lemley and in his own lab established that TOPO2 is a major factor stabilizing folded metaphase chromosomes. He really led the identification of components of human kinetochores and is responsible for discovering and cloning CNAP-B, CNAP-C, and discovered the inner centromere protein INCNP, which is very important in the process of chromosomes carrying stuff along with them during cell division. And really, Bill has established has proven the chromosome passenger hypothesis that chromosomes not only segregate DNA, but segregate a lot of the components inside the cell as well, not that we don't normally think of as being associated with chromosomes. And he's done work establishing the structural role played by SMC protein complexes in higher order chromosome structure. And today, he's going to tell us about a new direction, I guess, or somewhat new direction for his lab, which is large-scale proteomics for chromosome structure. And I think this material is soon to be published, but is very cutting-edge stuff, so I'm looking forward to hearing about it. Thank you.
Well, thank you very much for the invitation and the opportunity to present here. Yes, I'm going to explain to you what that means later. Spelling it out doesn't explain very much more at the moment, but the idea is to develop a strategy where we can, one of the things that we can do is we can study proteins that work in complexes, and we can study them in their native environment. In other words, lots of protein complexes do what they do when they're not soluble. And so we can look at that. And also, we're working on developing methods to predict the function of unknown proteins. And by that I mean unknown proteins who, when you examine the sequence doesn't tell you anything about the protein. So it's not just straight bioinformatics. So here's the structure of what I'm going to tell you about today. I'm going to talk about the discovery of the first kinetochore proteins. That's the first thing. And then I'm going to talk about these methods developing the complete proteome of mitotic chromosomes. Then we're going to go into the random forest, and we're going to wander around and get lost there for a little while. And then I'm going to focus on kinetochores, and I'm going to take all of this information and try to focus on what we've learned about kinetochores. What I'd like to emphasize, and I haven't put it on a slide, is the approach that I'm going to present to you today it's like a philosophy rather than just a set of experiments about chromosomes. So if you work on cell junctions or on any cellular fraction that you maybe can get your hands on but can't really purify, exactly this analysis would work for you. So I'm going to talk first about just a little history, the discovery of the first kinetochore proteins. And for those of you who don't work on mitotic chromosomes or on mitosis, it's an old subject. This drawing was drawn in 1904. It's a classic subject. The spots here represent kinetochores, the red dots. And here you see, this is a chicken lymphocyte. and it was a rather challenging cell to image. And you're seeing green is a protein called EB3 that surfs on the end of growing microtubules and then jumps off when they start to shrink again. Red are the kinetochores. And red is actually GFP knocked into an endogenous gene in this particular cell line. So the red things are little button-like structures on the surface of the chromosome, and here's an electron micrograph, and they're what the microtubules attach to, and they do lots of wonderful things. And they're one of the most complicated substructures in the cell. It's going to turn out that there are probably a couple of hundred proteins working in there, different proteins, I mean. So here we have the kinetochores are what mediates the attachment of chromosomes to the spindle. And as we proceed here, I can see I'm actually getting my passport back, which is going to be a good thing for me. I don't want to forget that. I love it here, but I want to go home when I'm done. And so the question is, what did this hand tell us about chromosomes? So now this is not a normal hand. This is a hand from a person who has something called Raynaud's phenomenon. And their hand has gotten cold, and all the blood vessels have shut off in their fingers. And so 24 years ago, I set up a collaboration to use autoantibodies from patients who have connective tissue diseases, rheumatic diseases, to study chromosomal proteins. And so when a woman like this would come into the clinic and she would see a rheumatologist, the first thing that would happen was they would take a blood sample from her and they would screen her for antibodies that recognized self, particularly nuclear antigens. And they used to use mouse liver for that, where you don't see any dividing cells. And there was a class of antibodies that was called a speckled nuclear pattern. And then a Japanese postdoc in the lab of Ing Ten, a guy named Moroi, put those antibodies on dividing cells. And he just must have thought, oh, amazing. Because it turned out that some of the speckles were actually centromere regions of chromosomes. They then kind of screwed up their biochemical analysis of this and were unable to identify the proteins. And so that's where we came in later on when I'd set up my own lab. Now to do this work, I set up a collaboration with Naomi Rothfield from Farmington, Connecticut. She's now, I think she's now retired. But she's a rheumatologist and was a really famous expert on lupus. But her husband was a really great scientist. And so he inculcated in her this sort of scientific attitude towards things. So she had, in the hallway, this line of -80 freezers, and in there were 65,000 patient sera. And what was actually really special and was not normal for a medical researcher at that time is those sera were all in a database. And she knew, so everything that was known about the sera was available to her. And we take that for granted now, but actually we started doing these experiments in the very early 80s. That wasn't the case then. So we found some quite special treasures in there, and I just want to show you one set of those treasures. So these are immunoblots. Each one of these is just an isolated strip that's been probed with a different serum sample from a particular lady who came into the clinic with muscular problems in the beginning. And just the particular type of symptoms she had, they thought, oh, she may be autoimmune. And these are actually artifacts, but she was making antibodies that were recognizing two proteins. Now, what's being immunoblotted against here are proteins from isolated chromosomes. So, and these were done in my lab. This is what they were doing. They just knew that she had autoantibodies. But they didn't know what any of this was. But now she kept coming into the clinic, and between this visit and this visit, she developed anti-centromere antibodies. So this means that now these antibodies are quite rare. Maybe about 5 or 10 per million in the population per year is the incidence of this. We have pre-immune serum from a lady. That's just amazing. And so now she developed the anti-centromere antibodies, which you could see by the immunofluorescence, and you could see that 3 new proteins were now recognized. And we called these centromere protein, or CIMP A, B, and C. So this was very exciting, and we went on to work with these proteins. And this is the serum from, actually it's from a woman named Grace Simpkins, who was a real hero, or a real heroine. If you have autoimmune disease, particularly if you have scleroderma means hard skin, and you have excess collagen deposits, and it means for you getting bled is incredibly painful and difficult. And this woman came in and volunteered to be bled for us. With human patients, people have to be willing to do this. So, and her serum is amazing. You can take this serum and you can dilute it 1 to 1 million fold, and you can still see a signal on bloods. So we used her serum to clone the first 2 kinetochore proteins, and we were really proud of this. We beat the yeast here. These were the first 2 kinetochore proteins in any species But now when you look at that, but those three proteins, it just looks so silly because this is now the budding yeast kinetochore in a fairly recent review, and you can see there's a huge number of proteins here. And actually, this doesn't begin to cover all the proteins that people think are at budding yeast centromeres, which is more than 90. And so this raises a question, how many more kinetochore proteins remain to be discovered? And if you're interested in the sort of the reductive approach to science, you want to make the complete list, and you want to figure out what everything does, but you need the list first. So this is actually quite an important question. So to aim in that general direction, but also to learn about chromosomes in general, we isolated chromosomes by a method that I learned when I was a postdoctoral fellow in Switzerland in Uli Lemley's lab. And this looks pretty simple. You lyse cells and you run a couple of gradients, and you have to use some specialized detergents. And it only takes you about a year to learn to do this well. And that's how long it took Shinya Ohta, 1 year of absolutely systematic experiments before he could get these chromosomes completely clean, no mitochondrial contaminants. And what you end up with is something that looks like that. And what I'd wanted to know for years and years was, what are all these proteins? We'd identified a few of them by making antibodies, but we didn't know what all the other ones were, and we really wanted to know. So the way you're going to identify all those these days is by collaborating with a mass spectrometrist. And I have to say that this has to be to do this kind of work, it has to be a full collaboration, because of the stuff I'm going to show you, you can't buy this in a facility, I don't think. And I'm also showing this because in case you have any questions about the mass spec, that represents my understanding of it. All right, so the first thing you always get when you do this is you get a list. And as you can see, it's a huge list. And I want to give Shinya Ohta, the postdoc, some credit, because these were chicken mitotic chromosomes for a reason that will become apparent later in the talk. And the annotation of the chicken database is not very good at all. So he ended up getting 3,500 unknown proteins. So he did 3,000 blast searches, one by one. He could do about 150 a day, and he just cranked through them. And figured out all but about 560 of the proteins. So that was a heroic effort on Shinya's part. Now you look at this. This is the democratic view of chromosomes. This is completely not representative of actually what's in the tube. This is one protein, one vote. So, and because this looks like the chromatin stuff, but here we have the histones, this little thin slice, because there's only 20 different histones that we found. And that's completely not representative of what chromosomes are, because chromosomes are made of chromatin, and we know they're full of histone. And these are nuclear proteins, which may or may not be real. There's a lot of really interesting information in here, which I'm not going to go into. For example, people who are interested in bookmarking of genes during mitosis, we know the list of all the transcription factors that are still there. There's a lot there. One thing that's important point to make is the centromere protein. So one thing you might ask, you say, that's a huge list. How thorough, how penetrant is your list? Every protein that's ever been identified and been said to be its centromeres we found in this, except for one protein that's not in the chicken database, and so we can't find it because we don't know what its sequence should be. All right, so we feel like this is probably a pretty good definitive start. Right. So what are we going to do? The way we're going to figure out what the chromosomes really look like now is we're going to do something which I'm going to gloss over, and if there's anybody who's interested in mass spec here, we can argue about this, which I suspect we'll end up doing at the end. We claim that we know how to calculate the absolute copy number of every one of those 4,000 proteins in chromosomes. And the way we're going to test that and what we're going to do with that, one of the things we're going to do with it is for each protein, we're going to take its copy number and we're going to multiply it by the molecular weight of the protein. And that's going to tell us what's the mass of protein, or what's the weight of that protein in chromosomes. And now you see the answer. And the answer is that chromosomes are mostly histone. In fact, when you measure this biochemically, it's 60%. But the number we're measuring by here, we're using H4 as one of the histone H4 as one of our standards. So that's several times 10 to the 7th. So we're multiplying absolutely huge numbers. And if we were off in the numbers, this would just blow up. But it doesn't. So chromosomes are. So this now makes the prep look pretty good. These are all the guys we know are in chromosomes. These are the ones that we're pretty confident aren't in chromosomes, and you see some other stuff there too. All right. But that's a lot of proteins. And so how do you figure out what's interesting? And in fact, how do you figure out what's really chromosomal? And that's a real big problem. And that's because of this. So this is a really great movie made by a guy named John Dahm, who works with Gary Gorbsky in Oklahoma. And this cell is about to divide. So actually now you can start to see the chromosomes are starting to condense. And at that point, the nuclear envelope broke down. And what happened? Well, chromosomes are incredibly highly charged. So when the nuclear envelope breaks down and they go into the cytoplasm, chromosomes are roughly speaking 1/3 DNA, which is extremely negatively charged. They're roughly 1/3 histone. It's a bit more than that, which is extremely positively charged. So cytoplasmic proteins just stick to them when they go into the cytoplasm. And if the proteins are stuck to them, then that raises a complicated issue of what's a chromosomal protein and what isn't. If a protein's stuck to the chromosome and you purify chromosomes, it certainly is a chromosomal protein. But is it functional? Well, that's an issue. So to try to answer this was a really vexing question. So to try to answer this question, we developed this thing that we call multi-classifier combinatorial proteomics. And I think the best way to start making some sense out of this gobbledygook is say, well, what is a classifier at least? So our definition of a classifier, which is not the definition that some machine learning people use, but it's the one that we like, is that a classifier is just a list. So the classifier is the output from one experiment. So and the way of thinking, it's just a list, and you want to make a list where the guys we're most interested in are on the top of the list, and the ones we're least interested in are on the bottom of the list, right? And the whole list is all our proteins. Like the copy number, the most abundant one is at the top, the least abundant one is at the bottom. It's just a list. Nothing fancy. Now, most of our classifiers use SILAC, which some of you probably know about, but some of you may not. It's a rather silly acronym. It stands for Stable Isotope Labeling with Amino Acids in Cell Culture. That's a technique developed by Matthias Mann. So to do this, what you do is you grow some cells in normal tissue culture medium, and you grow some other cells in medium which has heavy lysine and arginine. This is the simplest way to do SILAC. And we're going to mix the two populations of cells, and then we're going to isolate the chromosomes from them, and so we'll have heavy and light chromosomes. Then we're going to process those chromosomes the way we would normally process things to put them into mass spec, and now we're going to get spectra. And here's an example of spectra from just one peptide. And so this is the fragmentation pattern for one peptide, and if you know about the mass spec and you know how all this works, you can measure the difference in mass between these two guys, and you can identify that these peptides are the same as those peptides, it's just that these guys have C13 and these guys have C12, all right? Now, this is absolutely quantitative experiment because it's the same peptide run in the same machine at the same time. And so everything is flying exactly the same in the machine. And so I can take the ratio of the area under these peaks and compare it to the ratio of the area under those peaks. And that tells me the absolute ratio of how much of that protein came from these cells and how much of it came from those cells. This is really quantitative. All right, so I'm going to jump in with our classifiers. I'm going to jump in at number 3. And this is a SILAC experiment, but it's unlike any other SILAC experiment I think that's ever been done. So what we did what Shinya did, was Shinya took the mitotic cells, and these were light mitotic cells, and he broke them open, and he very gently just spun the chromosomes down just as gently as he could so he didn't bother them because, as John and his people know, chromosomes just love to stick to everything. So you don't want them to stick to each other here. Now you take the parallel culture growing in heavy medium, and you spin them pretty hard. So you have chromosomes and you have a post-chromosomal extract. Then you mix the chromosomes with the extract, and you incubate them. You let them go for a while, and now you isolate the pure chromosomes from them. And so what you're looking at here is we were initially calling this exchange, and it is looking at a way at exchange, but in fact we can only really look at the proteins that jump from here onto those chromosomes. We can't look at the proteins that jump off of the chromosomes into here because we can't really do meaningful mass spec on the entire cytosol. It's just too much junk, and Shinya threatened to kill us if we made him do any more blast searches. So that's that, and this is what it looks like. It works really beautifully. So here's a known chromosomal protein, histone H2B. It's all light. None of it came from the cytoplasm. Okay? And here's something we call a hitchhiker protein as opposed to a passenger protein, because this protein we don't think it's doing anything on the chromosomes because it's a ribosomal protein. And you saw this before, but there's about half of it from the extract and about half of it from the cells. So it's actually telling us some interesting things. This stuff jumped onto those chromosomes during the extract and stayed on tightly enough so that we went through all of the gradients and the ways we purify the chromosomes, it stayed on there, okay? But this stuff actually went on in the cell and stayed on in the incubation and the heavy extract and went all the way through. So these guys are really on the chromosomes for, they're on there for the duration. So we're pretty sure that a ribosomal protein is not really what we would think of as a chromosomal protein, but you're never going to get it off. Right? Okay, so that's what that classifier was. Now, another very informative classifier is just a way of saying how much of each protein is in chromosomes. So what you do is you isolate chromosomes and you make that same heavy cytosol, and then you just take you take equal protein masses of each of these guys, and you don't isolate anything. You just run that through the mass spec, and then you just say what fraction of every protein is in the mass spec. And so for this experiment, so here's what I'm talking about when you need your collaborator. This is a 5-day week, 24 hours a day on the mass spec, and I don't have any idea how much that would cost if you tried to buy it. All right. Now, another classifier, which is represented, so there can be 1 million classifiers, and luckily for you, we haven't gotten there yet. So this classifier is now introducing why we're using these chicken cells. And this represents a very important protein called in the condensin complex, and we're going to look at a genetic knockout of it. So condensin, this is the karyotype of it's an aneuploid karyotype of chicken cells, which have nice big chromosomes that look like human chromosomes, and all these little teeny guys, which birds have. These are actually little normal chromosomes that have got a bunch of genes on them. They're just little chromosomes. So what we can do with this is here's the condensin complexes, which is what's in sort of pink there. And this is a very important complex it has something to do with regulating the architecture of these chromosomes, but it's pretty fair to say that nobody knows what it does, and I know that John's lab is working on this. So in chicken DT40 cells, what we can do is we can knock out that SMC2 gene by targeted recombination, and we can then ask what's going on with the phenotype. So the question is, why would we pick these chicken DT40 cells? What's special about them? Well, it turns out that DT40 is a very powerful system for doing genetic analysis of essential genes. What they are is that it's a B-cell lymphoma line from chickens. Now, the way they grow, I mean, for people who work with vertebrate cell lines, the luxury of a cell that doubles population that doubles every 8 hours, this means that if you want to make stable cell lines, you have them in a week. This is really fast. These cells also do very high-frequency homologous recombination, so from 5% to 95% in the experiments in our lab. So this means that when we want, the movie I showed you in the very beginning where we had the red kinetochores going back and forth, we have a targeting construct that we used and we that was done by knocking GFP into an endogenous gene. That actually happens to be a single copy gene that's on a sex chromosome, and it's an essential protein, so we know it's a functional protein that we're making. And when we do that, 85% of the colonies that grow up that are drug resistant have the targeted gene. So it's almost as simple as, you say, why do immunofluorescence? Just knock this in. It's just going to take a week, and it's really easy. You can't do that with anything else. So because they're lymphocytes, they have big nuclei and little cytoplasm, so they're really great for isolating chromosomes. And these weird animals run around in the afternoon, and their internal muscle temperature is 45 degrees. So we actually have a TS degron system working in these cells where we can shift the temperature and make proteins go away within half an hour. But I'm not going to talk about that. That's just technology. All right, so here's a mitotic spread from the DT40 cells, and you see the SMC2 labeling cells. And if we now turn off, so the way we've done this knockout is we've targeted the endogenous gene, but we've also put into the cells an SMC2 cDNA that's regulated by a tetracycline regulated promoter. So when we add doxycycline, which is the more stable molecule, that shuts off transcription of this transgene that's keeping the cells alive. And a few days later, the cells are going to die. And what we're going to do is we're going to just, what Shinya does is very, very carefully monitor these populations of cells and get them just when the amount of SMC2 is starting to drop through the floor. Now, we can't look at cells that don't have any SMC2 because it's an essential protein and dead cells don't divide. Right? So, but you can see he's found a level where you really can't detect it. And we can't detect it biochemically either. So you've seen this kind of thing before. We're going to take those cells, the knockout cells, where we've added the doxycycline, and we're going to grow them in light medium so that they're happier. we're going to take the cells where the protein is still being expressed. It's actually the same cell line. It's just that in this case, we've turned off the rescue. In this case, the rescue is still on. We're going to mix equal weights of them, and we're going to isolate the chromosomes from them. Yeah. And then we're going to run this for a week on the Orbitrap. So now we come back to this fact. This classifier is just a way to make a list. So let's look at the lists. So here they are. So that's the abundance, and we have that for every protein. And you immediately see one of the bugbears of proteomic experiments, which is you never identify every protein in every experiment. So that's why these are rug plots, and that's why the rugs are shorter. All right, so that's going to be a problem, actually. Now, another problem is that the way we've color-coded things, we're interested in what's red, and we're not interested in what's green. And you can see immediately that there's nowhere you can draw the line to separate the red from the green. So this was a real bummer, actually. There's a lot of money invested in this. And I expected we were going to be able to combine these things and we were going to see something. And I can tell you that, in fact, this one, which is I'm going to come back to later, this one is worse than random. So that's really kind of a bummer. So what do you do about this? Well, you've invested too much money, so you can't go back. So what you think about it is actually, well, what if we just get these things up off the rug? So a classifier is just a list. So a list is just the axis on a plot, is another way of thinking about it. And nobody had ever done this before, but it just occurred to us. You can just plot things against each other that seem to make no sense. Like we can plot the copy number of each protein versus whether it's a chromosomal protein or not a chromosomal protein. Or how well it exchanges versus whether it requires condensin or not, or anything you can think of. All right, but this turned out to be pretty cool, actually. So here's an example of one of those plots. We're plotting the abundance of the protein against whether the proteins are chromosomal or not. And that's all the proteins. You obviously can't see anything. So we look down here at a little bit of statistics of what we've got. These are the guys we're interested in. These are the guys we're not interested in. You see there's 1,100 that we're not interested in and about 500 that we are interested in. But now what happens if we say, oh, let's just look at this is really green down here. There's 4,000 proteins down in here. And there's just a few others scattered up here. So let's look up there. So we're going to take that box. And now when we look at what's happened, the percentage of proteins we're interested in is doubled in that box. So that's really great. And the percentage of unknowns hasn't changed very much, actually. So this is a way to find areas. So we can use these plots, and we can identify areas where probably we're interested in the proteins that are there. But we can learn some other things also, because it turns out that if you look at protein complexes, they cluster. And so that would mean that if I have this complex here and I have an unknown protein in there, I would say, well, that protein's in about the same stoichiometry and the same behavior as that complex. Maybe it's got something to do with that. We didn't think about this at first, but actually it's just obvious. If it's a 1-to-1-to-1 complex and we've calculated how many copies of the proteins there are, it's going to be a line like that, right? And if it's a protein complex, it's either in chromosomes or it's not in chromosomes, so it's going to be a line like that. So in these analyses, by plotting things against each other, we can get lots of interesting functional information, which I'm not going to actually explore more today. Now we have to deal with the fact of having the missing proteins and trying to figure out a way. What we really want out of all of this is we want to have a list of all of our proteins. It's a reliable list where the guys on the top of the list are the ones we really are interested in, and we've somehow managed to segregate that list and get the ones we're not interested in to the bottom of the list. So we're going to do this by machine learning. The method we use is called random forest analysis. And to do this kind of machine learning, you have to train the program to it's what you're interested in. So you need to pick training sets. And so we're going to pick. This is actually an older version of the histogram, but we're going to pick the red proteins, which we are very confident are chromosomal, and we're going to pick a subset of these green proteins, which we're very confident are not chromosomal. And we're going to train the computer to separate those sets. And so we're going to separate it. Basically, you build these trees where it's a whole series of decisions that get made, and the data get run like that. And that's the training data. All right? And now you take the wild data, which is the rest of the data that you haven't trained, and you run it through the trees. And then, but the problem is that each one of these trees is statistical, so it's going to make mistakes. And so now democracy comes into play. And so you can just calculate how many trees you need. In our case, it's a few hundred trees. And then you just take a vote. So if you make 500 trees and 100 trees say that protein is not chromosomal and 400 say it is chromosomal, you say, okay, it's chromosomal. That's the way you give it the score. And just so you can see what it actually is like, for a non-mathematical idiot like me, this is really nice because it's actually not a big complicated formula. It's just a decision tree. So you pick a protein and you come to a node, and the node is just a class. It's one of the classifiers that we made, and it's picked at random. That's why it's called a random forest. And then you just use a criterion and you decide whether you go left or right. And then this node is picked at random again, and the same protein you make, you have another decision that you make, and each one of these nodes gets picked at random. And you end up separating the proteins down this pathway, and the goal is to separate your training set, and it works. So that's what happens. This is the rug plot that comes out of that. I mean, it doesn't look remarkably different from these, but it's considerably enriched, in fact, in interesting things over on that side. And I'm going to come back to how we go beyond that. But what we had to do next is we had to validate this procedure. So what we decided to do, well, initially we decided to clone 20 proteins and tag them and see where they were. And then we submitted the paper to Cell, and the referee said, well, you cloned a bunch of proteins that you thought were interesting, and of course you found a bunch of proteins that were interesting. You have to clone a bunch of proteins that you think are not interesting and show that they're not interesting. Thank you so much. So that's what we got to do. And because nobody but us cares about chickens, what we actually did is we found the human homologs of all of these guys. And so we cloned 50 human homologs, tagged them with GFP, and we expressed them in human cells. And then what did we find? Well, we got 13 new proteins that associate with kinetochores that are not previously characterized, 7 proteins that bind on the periphery of the chromosomes like this, which is quite fascinating and not at all understood, and 13 other proteins that seem to be all over the chromosome. So we got lots of proteins, lots to work with. Shinya's goal in his postdoc was to find a new kinetochore protein that he could take back to Japan and start a lab. So his goals are changing. Now, what Shinya did then was to realize, and this is actually, I think, the biggest breakthrough, and this is Shinya's alone. No PI was involved in this. He wanted to learn programming, so he taught himself R. And then he found a series of programs that were written to quantify microarray data and to compare microarray data. And he said, well, yeah, fine. What if you run proteomics data through those microarray programs? So as far as I know, it's never been done before. And the analysis is what's called clustering analysis. It's just a way to see, it's a way for looking at proteins or transcripts or whatever that are going to co-vary in your different experiments. And you're not supposed to be able to see that. You can just see that they're blocks of color. And so if you look down here, so here you see a block of things that are all red. These, APC 1, 2, 4, 3, 6, 8. These are the subunits of the anaphase promoting complex. It's a very important ubiquitin ligase that's regulating it's regulating many things in the cell cycle. And one of our unknown proteins is sitting right in the middle there. So we predicted that this protein would be an APC component. We set out to try to test that. But while we were testing that, and luckily after the paper had already been submitted, a paper came out in Science which did a huge proteomic screen and a protein-protein interaction screen and identified that protein as APC 16. So this protein has no recognizable sequence motif of any kind, but you could predict from its guilt by association in this cluster what it was, and it is what it was. And just so you can see what that protein looks like, that's a marker for real kinetochores. And here's this protein. So you see most of it when it's tagged with GFP is all over the cells. And if we blow things up, there's bits of green at every kinetochore. So there's a population of this protein which is at kinetochores. And actually, if you knock the protein down by RNAi, in a control experiment, everything looks perfectly fine. But in a knockdown experiment, the cells look very weird and they have problems in mitosis. So this protein really does, its function really is required for chromosome segregation, and it looks like it's doing something at kinetochore. So that's a bit of functional data for that. All right. Well, we don't know very much about that protein, but now let's look at another one here. So we'll go in here, and we found a protein that we had heard of. And it was next to another protein. Well, actually what happened is we tagged this protein, and this actually gave us a beautiful signal in kinetochores. And it was next to another protein called SCA2. And so our prediction was that, that this protein would have something to do with SCA2. And forget that, because you don't have enough information to know why we would say that. So what we did is we decided to do a little biochemistry, and if you have access to a good SILAC collaborator, a good experiment, your pull downs become incredibly simple, because here's, when you normalize by all the junk that was in the control pull down, just 3 proteins pop up as being completely specific. And SCA1 and SCA2 were published as being in a complex, and so this protein is clearly in that complex with SCA1 and SCA2. And at the time that Shinya did this paper and did this experiment, and the paper had its first submission, which was to Science, this one paper on SCA1 and SCA2 was the only paper that had been published. Then in the next 6 months, 5 papers came out identifying this protein as being in a complex with them, and our paper got rejected at Science. So Shinya was not very happy about that, and neither were we, actually. But a collaborator of ours named Tatsuo Fukugawa from Japan had already had known about some of those papers, and he had isolated a knockout of SCA3 in the chicken DT40 cell line. So we have now, so this is another classifier, but this is a very specific one, because now we specifically knocked out one component that's involved in the kinetochore. And the question we want to ask with this kind of analysis is, what can we learn from that? I mean, can we use this kind of analysis? If we knock out one component of the kinetochore, is the whole chromosome going to be screwed up, or are we going to see something meaningful? And here's where we discovered that we can really learn about protein complexes. So we're going to look at, these are the ratios. So if the bar goes down below the line, there was less of the protein in chromosomes if it didn't have the SCA protein. And if the bar goes up, there's more of it. And this is most of the kinetochore proteins and main complexes that we know about, and nothing's happening, okay? But this is the SCA complex itself. So if you knock down SCA3, the other guys of the complex are also not in chromosomes. And if you look over here, this isn't sorted randomly. This is APC 1, 2, 3, 4, 5. This is all the subunits of the APC, including this guy, this bar right here, which is our new APC subunit that we had identified. And so the anaphase promoting complex is normally associated with chromosomes, and if you don't have the SCA complex, the whole complex, every subunit changes just the same. And over here, that's, again, not 3 random proteins. That's a protein called RANBP2 and RANGAF1, which make a complex that associate with kinetochores. And this is a sumo E3 ligase, and so we looked at UBC9, which is the E2, and it behaves exactly the same as them as well. So so nothing has been isolated, but the members of complexes are doing the same things. And that you can even see that here, where if you looked along this line, the most boring region of it is right here, where there's nothing happening. Actually, that's all one complex. It's a nuclear pore complex that is at kinetochores. So if we had knockouts in other genes, what we could do is knock out a whole series of genes and then look at all the complexes that change. And in fact, we do, because these are the knockouts that Tatsuo Fukugawa has made available to us. So what my PhD student Laura Wood is going to be doing is going through and looking at all of the complexes represented by these knockouts and determine a pathway for looking at the kinetochore. It's assembly of the kinetochore. Now, the blue guys that went up over here are the mitotic checkpoint signaling proteins. And there's something interesting also to be learned for this, which is a little bit for specialists, but maybe not. And that is that there's a complex of MAD2 and BUB-R1 and a protein called CDC20, which it's called the mitotic checkpoint complex, and that's supposed to be very important in signaling. And the checkpoint complex inhibits CDC20, but what CDC20 does is CDC20 associates normally with the APC, and it sets its specificity for the targets it's going to hit. And so you could say, well, when those guys are on chromosomes, what is CDC20? Is it a member of the mitotic checkpoint complex, which is, I think, what everybody thinks it is, or is it a member of the APC? And that's CDC20 right there. So it's pretty obvious. It's a member of the APC, which is on chromosomes. All right. So now, where this analysis gets really exciting, it came to me when I was sitting in a seminar where I was struggling between falling asleep and thinking about other things that I had to do, and grumbling in my mind about comments from referees on this paper. And I realized, actually, that this combinatorial method, there's no reason to limit it to proteomics. So anything where you can come up with a list of numbers describing your proteins, you can put into the mix. So you can make a real mixed salad here. So that's what this shows you. So this is our training data set. Red is what we're interested in. Green is what we're not interested in. The first thing you can see is the random forest analysis is incredible. We can really, there's one red spot to the left of this line, and there's 2 green spots to the right of that line. So we have an almost perfect separation of our training set. But the way we would draw this line is because it's two-dimensional. So this is just the random forest here, that rug plot that you saw. But now here we've added in another classifier. And what this classifier is that Jimmy Carlo, who's a postdoc in Yuri Rapsilber's lab, took all of the proteins that we identified, and he wrote a script to look for recognized protein domains. They were annotated in protein domain databases. And so he identified all the easily identified protein domains in all the proteins that we found. And then he said, well, okay, for domain X, how frequently is that found in a red protein, and how frequently is it found in a green protein? So is it a chromosomal domain, or is it a cytoplasmic domain? This is way too simplistic, right? But you can just say, statistically speaking, does it tend to be more in one than the other? And then he gave every domain a score. And so that stretched the material along this line, and it actually increased the separation so that when we plot this, just all our proteomics against the proteomics plus the analysis of the domains, now we get this almost perfect separation of our data. The sorts of things that you could add into this, if you wanted to add in more, is you could do a microarray experiment or go and look up online, anywhere you can find access. You could add in microarray expression data here. You could look at different cell types. If you had RNA, if you looked at one of these big RNAi databases where they'd RNAi'd the genome of Drosophila or something, and you found all the homologs here, you could take the mathematical scores from their database and include all them. So any kind of mathematical analysis that you do, you can include on an equal basis. And that's, I think, what's really new about this analysis, because you can integrate everything. So the question, one of the questions that I asked back in the beginning was, how many new kinetochore proteins are there? We started with 3, and we've now gone beyond SIMP-Z. And I was really hoping, actually, to use On Beyond Zebra by Dr. Seuss. But it turns out you couldn't use On Beyond Zebra because they were these long sentence, silly names. So we go from Z to 27, 28, 29. We decided we'd stick with the English alphabet. And all right, so the random forest, though, can tell us how far there is to go. And it turns out there's a long way to go. So this was our 2D analysis where we were able to separate our training set. Well, this looks wonderful, but it's less wonderful than it looks because it has to look like this because we trained it to look like this. If it's well behaved, if it's not a typical PhD student and it's very nice and well behaved, it'll separate like this. So now let's look at how the wild proteins look. And that's over here and down there. So the gray things are unknown proteins that we don't know anything about. And every colored symbol here is a protein that we know about because we cloned it and we asked where it was. And so that's centromere. These are chromosomal. These are around the periphery of chromosomes, and these are unlocalized. And so if we look, so we can look to the left of the line and to the right of the line. To the left of the line, we have 14 boring proteins to us. They may be very interesting to almost everybody else in the audience. And then 2 proteins that we're interested in. So, and then if you look to the right of the line, you can see that we have 30 proteins that we're really interested in and 4 that we're not. So, in fact, it turns out that we get about a 90% ability. 90% of the proteins over here are proteins we're interested in, and 10% of the proteins that are over there are what we're interested in. So we get a really, really good separation of what we want. So, if this analysis, if we really did clone the 50 proteins in an unbiased way, and we were pretty unbiased, although to be honest, we didn't clone the proteins whose sequence were predicted to be 300 and 400 kilodaltons. We cloned the proteins that were predicted sequences to be small. Then we should be able to say, okay, there's 224 unknowns here and 287 unknowns here, and what's the percentage of guys we're interested in down in the histogram? That says there should be, in our data set, roughly another 100 or so centromere proteins and a bunch of chromosomal and periphery proteins. So there's a bunch more interesting proteins to find in our data set. And I think this is pretty unbiased prediction for that. So Shinya is not going to have to settle for one protein. He can just work for the rest of his career on that, I think. So what we did here is we developed some new approaches to analyze the complex proteome of the chromosomes. And it wasn't just like you just needed one more column or one more trick and you could get them more pure. I think we got them as pure as you can get them, but you still needed to figure out what it meant. And along the way, we cloned a bunch of new chromosomal proteins, and that's going to keep a number of people very happy for a long time. And using the analysis I showed you with that kinetochore protein, we can do assembly pathways. So this can be done for anything. So this is the advantage of having the genetics to mix in with the proteomics. And we can predict how many more proteins remain to be discovered. So that's pretty exciting. Now I just want to jump back and go back to what I kind of glossed over, but I know that John was interested in this earlier, and some of you might be. So we were able to use a spectral analysis, which was modified. The way it was done was modified by Yuri and Jimmy Carlo, his postdoc. And we can calculate. So at telomeres, we calculate there's about 20 copies of the complex that shields telomeres. Whereas at kinetochores, there's 12 nucleosomes that are the specialized nucleosomes where this protein SIMP-A replaces H3. And this seems to be the apex of the assembly pathway to make a kinetochore. The most abundant non-histone protein on chromosomes is topoisomerase II, which I think John's people will be very pleased to hear. But the second most abundant protein, this is a complete shock to us. It's an enzyme, poly-ADP ribose polymerase. It's the second most abundant protein. We don't know anything about a single substrate for this protein. But what we do know is that when it converts NAD into ADP ribose chains, those chains are at least as negative as DNA. And in fact, they can work extremely well in remodeling nucleosomes. So this is probably quite active. It's probably doing something. If we look at the checkpoint complexes, this is the signaling complex. There's 17 copies of that. And if we look at the interface promoting complex, there's 17 copies of that. Now, when I say 17 plus or minus 9 here, we identified this complex has 16 components, but actually they're not all annotated in the chicken genome, so we only could identify about 8 or 10 of them. And this is the copy. Each one of those, we calculated the copy number independently. So 17 plus or minus 9 is the range we saw over that whole thing. So even if one protein's peptides flew weirdly in the mass spec, which people who know about mass spec will say that, that's why you can't do this copy number, we're averaging things out here. And so we're looking at a bunch of proteins. This guy we looked at all on its own. So how could we test that calculation? In a collaboration with Christina Flores in Edinburgh, what we did is we did palm microscopy. So that's photoactivation localization microscopy, which is it's a form of microscopy where you can look at well, we were using 37 nanometer resolution for straight observation, and it works by having fluorochromes blink on and off. And actually, from the number of blinks at particular locations, you can calculate the number of copies of what you're looking at. And her calculation from palm microscopy, which I think from my very quick explanation couldn't be more different from this. Her calculation was 25 to 40 copies. So this is 24 copies because there's 2 per nucleosome. And her calculation was 25 to 40 copies of this protein. So it couldn't be, that's pretty damn close. We would be very happy with an order. We'd like to be better than an order of magnitude, but certainly we certainly wouldn't claim better than twofold. And the last thing here, you see the skin around the chromosomes. Those are incredibly abundant proteins, and they're mostly coming from the nucleolus. And are they just along for the ride, or are they doing something to help protect the chromosomes in mitosis? I think they may be helping to protect. So we'll kind of close out here with the great kinetochore face-off. And the question is, we're going to look now at the difference. We're going to use 2 different techniques. Well, actually, one of them is going to be used by Ted Salmon in North Carolina, fluorescence microscopy. And we're going to compare yeast kinetochores and vertebrate kinetochores. And we're going to ask, how many copies of a number of the centromere proteins are there per microtubule that binds to the kinetochore? Because the budding yeast has 1 microtubule that binds to the kinetochore. Humans have 20, and chickens have 4. All right? So that's the budding yeast data. So this is number of copies, so 2 to 8 per kinetochore bound. That's budding yeast data determined by fluorescence microscopy, and proteomics data determined from vertebrate cells gives you that, which says, well, these guys look like they're really organized probably the same way, even though actually, many aspects of kinetochore assembly would make the budding yeast kinetochore. People, I think, traditionally thought, yeah, the same proteins are there, but the organization is probably different. But probably it's not different. All right, so now where do we want to go with all of this? Well, we want to eventually end up producing a map like this for the kinetochore. And that's our goal over the next 5 years. And this is a fanciful drawing by Graham Johnson, which is based on a number of actual crystal structures and things, but altogether it's a fanciful drawing. So how are we going to do that? Well, we're going to do that in our collaboration with Yuri, because Yuri Rapsilber, in addition to being a guy who knows how to take the mass spec apart and put it back together and has developed all these other methods, is also developing a new method for structural analysis. And so what his analysis, we believe, within 5 years is going to enable us to do is to take isolated chromosomes, fix them, fix them with a chemical cross-linker, fragment the proteins, isolate the cross-linked products, and identify every peptide that cross-links to every other peptide in the entire chromosome. And this is very complicated analysis to do, and it's computationally incredibly difficult. And Yuri's he's a pretty special guy because his group is probably 60% informatics people, and that's not sort of bioinformaticians who kind of failed at doing experiments. These are hardcore computer guys. And so if you take a peptide fragment and you put it into E. coli extract and you run it on the mass spec, and then you want to get the sequence of this fragment that you now pull out in the mass spec, you have to do 10 to the 9th search operations. But if you take 2 peptide fragments and they're cross-linked together, you can actually model them as a linear thing. So the fact that they're cross-linked isn't a problem. But this guy could be cross-linked to every other conceivable peptide in there. So the number of search operations now goes up to 10 to the 12th. So this meant that the first time they did this on the, I think, 1,000-core machine in Edinburgh, it was a 6-hour search to get 1 peptide. Now, 2 years later, it's 0.3 seconds. So we're on the way to doing that. And when the chemistry is perfected, and Yuri has developed a cross-linker that can also be isolated, although kind of amusing story about that, for the mass spec people, the first cross-linker he developed that you could cross-link and then isolate, it turned out that cross-linker is an amazingly potent inhibitor of trypsin. Trypsin is the enzyme you have to use to fragment the chromosome. So this was really amusing, actually. But we're on the way. So I think we're going to be able to have that kind of a model in 5 years' time. So we did all the stuff you did before, and I think that it was close by saying the future is looking pretty bright here in Scotland. And I need to tell you who did the work. So this guy is an absolutely fantastic, creative, hardworking, careful postdoc. Yuri is really he's going to be one of the world's great mass spectroscopists. Jimmy Carlo is his postdoc who did a lot of the random forest analysis. And Tatsuo Fukugawa in Japan is our collaborator who's just an amazing genius at making knockouts. And I thank you for listening, and I have to thank my sponsors who are the Wellcome Trust. So thank you very much.
Questions, if anyone has any questions? Jason?
So I'm very curious about So I'm very curious about the classifier idea and the notion that you could take basically any classifier and it would add to the distinction between a chromosomal and non-chromosomal protein.
Yeah.
It doesn't really make sense to me that that would be the case because there's got to be classifiers that are completely irrelevant.
Well, yeah.
Or confusing. So if you plot chromosome or protein size versus abundance.
Yeah, we haven't done something like that.
To just, presumably you're picking classifiers that make some sense.
It's a little bit of this analysis that's like what, for a postdoc of mine who developed an image analysis program, and when asked how it works, he said, well, actually it's an alien intelligence. I don't know how it works. I know what it does. So the classifier number 5, which was the knocking out the kinetochore protein and looking what happened, actually when you looked at the data ordered there, it was worse than random. The really weird thing is there's a type of analysis that you can do if you're into the statistics of this stuff called receiver operating characteristic analysis, which is sort of sensitivity versus specificity. And it's a rigorous mathematical way. You can say, if I get if I want to get 100 chromosomal proteins out of your mix, how many junk proteins am I going to get with it? And the really weird thing is, so this classifier is worse than random. If you add it to the other classifiers, it makes them get better.
But that's not a surprise, because worse than random means if you multiply its output by -1, it's predictive.
Okay, well, see, I failed math everywhere along my career.
Do it.
Okay.
So that's good, not bad.
Okay.
Worse than random is predictive. Yeah, right.
Do you want to ask a question?
No,
okay.
Just giving me a lecture here. That's fine.
That's fine. Good. Any other questions? I was going to ask the complex identification aspect of this looks very powerful. had you only looked at this one SCA3 knockout?
Well, we have all the other knockouts in the lab. And,
Yeah, right.
You know, we're trying to get the first paper published.
So I was curious if you had data yet for SMC component knockout, because
Well, we did have that. So that was the SMC that was the SMC2 knockout. And so what we could see was every known subunit of SMC proteins was basically gone from protein. Maybe I didn't show that, but
I don't think you showed that slide. Yeah, okay.
well,
so I was curious.
I mean, I can show
curious
you
if you could
so
see.
every SMC subunit is gone from chromosomes,
Yeah.
and a bunch of other chromosomal proteins actually go down,
Yeah.
and a bunch of the crap proteins go up. And that's because the SMC2 knockout chromosomes are a little bit bigger and a little bit fluffier. Probably they've got more surface area, so more junk sticks to them. So yeah, so SMC2 we've done, and this kinetochore protein we've done. But Shinya at the moment is growing cells to look at cohesin knockouts, and we have a huge long list. And the thing is, though, that it's true that if in future experiments one might want to do more controls, but we're kind of sticking with what we know works. But so we can link up with toxicologists. So if you have hexavalent chromium or some compound that causes aneuploidy, there's no reason why we can't treat cells with hexavalent chromium, determine the proteome, and put that into this mix and identify all the proteins that vary based on that. And the reason we need all the classifiers is we need all the classifiers to build a random forest so that we can know which proteins are the ones that we're interested in. Because we've got all these proteins on the list, but we're not equally interested in all of them.
Very good. Okay. Any more questions? Tom. Oh, as a PSOC member, you're obliged to use the microphone.
So there's some 300 odd proteins at least in the chromosomes that you're pulling out, or in the centrochores. And how many of these do you think in the end of the day are going to be doing work, or catalytic, or signaling? And how many are kind of really structures and buttresses and flying arches and stuff?
Yeah, that's a really interesting question. Another way to phrase that question is how can we have the temerity to say that we're going to identify another 100 kinetochore proteins when people have done I don't know how many genetic screens in Saccharomyces and Schizosaccharomyces and looked in C. elegans and everywhere else, and they've identified all these proteins. So why should we be identifying so many more kinetochore proteins? So to answer that, then I think the most meaningful answer to that comes from our collaboration that we have that I didn't talk about with a bioinformatician at Oxford named Chris Ponting. And what Chris is doing is just he's trying to figure out what he can find out about the most unknown of the proteins where there's really nothing known. And what he's coming up with those is about half of the new proteins that we're seeing associated with centromeres in mitosis are involved in tinkering with chromatin. So there are various sorts of proteins whose domain structure, they're not studied, so we don't know for sure, but their domains tell us that they've probably got to do with chromatin modification. So probably they weren't picked up as kinetochore proteins because when you did something to them, you probably screwed up the chromatin and maybe killed the cell or had other pleomorphic phenotypes. So we're probably picking up proteins that are pleomorphic, but maybe part of their job is at the kinetochore. I think most of the really basic building blocks have been identified.